Hi everyone, my name is Elizabeth Bingston, and today I'm going to discuss adaptation of the ELISA to measure immune complex degradation in lupus. And so to begin, I'd like to discuss systemic lupus erythematosus, also known as SLE. SLE is a complex autoimmune disease. It affects about 1.5 million individuals in the United States alone. It causes systemic inflammation, and 9 out of 10 individuals with lupus are women, so it's predominantly in women. Like a lot of other autoimmune diseases, SLE has flare-ups and times where the symptoms are more severe than other times. And one of the major complications of lupus is lupus nephritis. It occurs in about half of patients with SLE. And in the lower right, that picture is a kidney with lupus nephritis. And currently, the primary treatment is using hydroxychloroquine, which is an immune suppressant. And so some of the symptoms of SLE include systemically a low-grade fever, photosensitivity, inflammation in the lungs and the pericardium, arthritis, fatigue, loss of appetite. But one of the most distinguishable physical symptoms is the butterfly rash along the cheekbones and the nose. It's usually pretty indicative of someone with SLE. And so diagnosing SLE, so the first thing that they look at is they do an anti-nuclear antibody test. And so anti-nuclear antibodies are autoantibodies that attack the nucleus of your own cells. And so if this test ends up being positive, the physicians will go through and apply the additional criteria. And so it's split into clinical domains and immunological domains. And so how they classify it is you have to have a score of 10 or more. And so within each like section, so within like neuropsychiatric, they're going to take the highest weight. So if a patient has delirium and seizure, they'll take the 5 points for seizure and then add it all up. But my research primarily focuses on the SLE specific antibodies and more specifically the anti-double stranded DNA antibody. And so immune complexes SLE. So when a cell is undergoing apoptosis, it's going to release microparticles into the extracellular space. And dendritic cells and macrophages are going to sense that this microparticle is in the serum. And so they secrete an enzyme called DNase 103. DNase 103's whole job is to go degrade the chromatin in these microparticles or around the microparticle. And when this happens, there's no autoimmune response. Your B cells don't target this as anything foreign. However, if DNase 103 is mutated or deficient, these microparticles are remaining how they are and the chromatin is not being degraded. And so your B cells are going to think this is an antigen. And because of that, they're going to produce anti-DNA antibodies. And so these will go and target these microparticles and form immune complexes. And these immune complexes are really prevalent in SLE, and they're really detrimental because they go and deposit into the kidneys, the lungs, the heart, and can cause different problems and complications. And it also leads to inflammation. And so DNase 103 is an endonuclease. It does cleave DNA. It acts extracellularly and intracellularly. It prefers to act extracellularly and will degrade the chromatin in the extracellular space, but it can go into the microparticles in those cells and degrade chromatin if it needs to. When the DNase 103 fails to degrade microparticles, it leads to elevated serum DNA, which is why these autoantibodies are being created. Children with mutated DNase 103 develop SLE, and DNase 103 might chew up the whole immune complex. It might just chew up the chromatin, but if it does chew up the whole immune complex, it's going to reduce pathology. And this is a structure of DNase 103 and has a signaling sequence, the nuclear locating sequence, and the CTH. And the CTH is really important because that's what allows the DNase 103 to degrade the actual chromatin. And so my hypothesis is DNase 103 degrades immune complexes, thus reducing the inflammation during SLE. And so to kind of lead into my assay that I perform for this, there's 3 different types of ELISAs. There's the direct ELISA, the indirect ELISA, and the sandwich ELISA are just a few of the types. Direct ELISA, you're going to coat your plate with your antigen. And then you're going to add a primary antibody conjugate that will have the specific enzyme. And so when you add substrate, it's going to cause a reaction and produce a color change in your wells. Direct ELISA is often used for testing antibody affinity. You're going to know how much of the antigen you're coating your plate with, and then you can see how much of the antibodies like bind to the antigen. And then there's also the sandwich ELISA. So you'll coat initially with a capture antibody, and then you'll add serum with antigen, and then you'll have your primary and secondary antibody, and it'll do the same sort of color change reaction. And sandwich ELISAs typically you don't have to purify the serum, so it'll tell you if this antigen's in the serum or not. And then the indirect ELISA, you're going to coat with an antigen just like direct, but instead of only having a primary, you're also going to have a secondary antibody conjugate that binds to the primary, and will also produce a color change when you add the substrate. And indirect ELISAs can determine the antibody concentration in a sample. And so that's pretty much what we're doing with the assay, that the immune complex degradation assay, because we're looking at how much immune complex there is, and that relates to how much antibody there is. And so the immune complex degradation assay, like I said before, is inspired by the indirect ELISA. And we're wanting to know, does DNase 103 chew up these immune complexes? And if it does, how much is it chewing up? And so we're testing the percentage of immune complex degradation. And so to just pictorially show y'all, so we add the antigen to the plate. So we add the thymus DNA to the plate, and we add the same concentration throughout all the wells. And then we're going to add 1% BSA and PBS as our blocking buffer, just to ensure that we have specific binding between the DNA and the primary antibody. So then once that happens, we then add our anti-double-stranded DNA antibody, or primary antibody. And so that's going to bind to the DNA. And that is the same thing as an immune complex. So we just made our immune complex. And so unlike the indirect ELISA, in between steps 3 and 4, we're going to add DNase 103. And we do that in a serial dilution. So we dilute it serially in the wells. And then so the DNase 103 is going to go chew up the DNA. And so when it chews up the DNA in the wells, it's going to release the primary antibody into the serum. And so when we wash our plates, if the antibody is not bound to the DNA from the well, it's going to be washed out. And so that's how we're able to tell how much of the degradation is occurring. And then we'll add our secondary antibodies, the anti-mouse HRP antibody. And so the HRP is the enzymatic part of the antibody that's going to react with the TMB to produce a colored product. And that colored product allows us, so we put the plate on a plate reader, and the plate reader is going to give us back absorbencies based on the colored product. And so this assay was completed and determined that 5.7 micrograms of DNase 103 results in 42% degradation of immune complexes. And so this is the graph on the left is the standard curve. And so in the first 2 columns on the plate, we did not add any DNase 103 because we wanted to just form immune complexes and be able to see what those absorbencies are at. And so we know how much we added of the anti-double-stranded DNA antibody. And so this is going to tell us the relationship between the amount of immune complex and the absorbency. And so we use this equation that was produced in order to determine how much immune complexes were degraded, because in the other 4 columns on the plate, we did add DNase 103, and it did, well, we're hoping it chewed up the DNA, and we don't know exactly the concentration of those, but we do know the absorbance, because when we put it on the plate reader, it gives us back the absorbance, which is Y. So then we're solving for X, which will tell us how much immune complex is left. And so then we're able to find the percentage, because we also do a control well where we don't add any DNase 103, so we can compare the amount that's degraded to the amount that should be there. And once we did that, we were able to know that when we had 5.7 micrograms, degraded about 42% of the immune complex. And so as we decrease the amount of DNase 103, the amount of immune complex degradation also decreases. And this helps support the hypothesis that DNase 103 is degrading. these immune complexes. And so in conclusion, these assays are going to help verify that DNase 103 is chewing up immune complexes. These immune complexes cause problem in SLE, and so preventing them could reduce pathology. So it's important that we're studying this. And DNase 103 can work in vitro, so it's worth proceeding in vitro and continuing to do research. And so some goals for the future is to perform the immune complex degradation assay with both DNase 103 and DNase 1. It's going to help verify that DNase 103 is the only enzyme able to degrade immune complexes and contribute to the symptoms of SLE, and that DNase 1 does not degrade these immune complexes. And also some goals are able to use different animal DNA and eventually human DNA to test immune complex degradation, because currently we are using calf thymus DNA. And I'd like to acknowledge Dr. Kyle, Minal, and Gavel, and all the members in the Kyle Lab for supporting my project. Thank y'all for listening.